Good morning. It is my great pleasure to be with Professor Marlies Knipper and Professor Lukas Ruettiger and Dr Wibke Singer from Tübingen Hearing Research Centre. And they joined me this morning to talk about their research studies and some of them that they want to present in the 6th International Conference on Hyperacusis and Misophonia on 1st and 2nd of July in London. So thank you to all of you to joining me. And Marlies, would you like to start and give a bit of an introduction to the research studies that you want to present in the conference?
Yeah, so with pleasure, Hashir. I will give you, as those people who probably have a lot to do with people with tinnitus and hyperacusis, an overview about the recent studies. And while in the previous years we focused a bit on tinnitus, we now focus much more on tinnitus with and without hyperacusis, hoping to learn a lot about hyperacusis. And what we will do is give you a brief overview about, meanwhile, 3 different studies who analyzed the patient groups of different ages and looking to, let's say, objective functional biomarkers, which hopefully you in the future may have the chance to use in clinical diagnostic. And indeed, we are happy, and this is what we will tell about, to have identified some of these objective markers independent of audiometry or fine structure of audiometry, also through imaging studies and independent of questionnaires, which we typically use. And I will not tell so much about the results, but we at least were surprised to learn how important it is to differentiate between the groups and how much we may have the chance to learn about hyperacusis. And if we are lucky, we may have the chance to talk about, to differentiate all these 3 groups.
And I know that you have been working on these biomarkers of tinnitus and hyperacusis for a long time. I remember many years ago they were focusing on animal studies, but now it seems that you're doing studies on humans as well, on patients.
Yes, this is correct. So we started to do this on animals, where we differentiated tinnitus and then later on also differentiated tinnitus from hyperacusis. And what we did not expect, indeed, is that our new findings support really the notion that it's possible to use at least some functional studies in animals as well as in humans. And we can confirm the first time our long-lasting studies in animals, which over a long time created a lot of irritation in the neuroscience scene, because they could not be confirmed by a lot of people. And now we learn, maybe, and this should make you perhaps curious, maybe the animal studies, or a lot of animal studies which were used since years, identified hyperacusis, but not tinnitus. But I leave you with this. We will take this and discuss this with you.
Oh, fantastic. Lukas, what do you think? Is there anything that fascinates you and you'd like to share?
Of course, I have to admit, it's always about the humans. So all our research that we always did that seeded actually more from the tinnitus problems than from the hyperacusis problems. The idea was always that it is not understood in the humans what really are the mechanisms of the development of these diseases. And this is what led us to these fantastical already at that time existing animal models on tinnitus. And this enabled us to find many molecular threads and molecular origins of the disease, how that lead to the development of tinnitus. And it was only later, more or less accidentally, when selecting animals for their behavior in after tinnitus induction, that we noted that they split up in different subgroups. So as Marlies already said, that they partly also developed hyperacusis, and these behaved completely different. And also the molecular markers and the functional markers that we found in these animal models were different. And this is what brought us back also to the human studies, because the high diversity in the human population that suffers from tinnitus and particularly also hyperacusis is not a homogeneous group. So we have the problem that we need to diagnose very exactly what is the impact of the tinnitus and what is the impact of the hyperacusis in a certain situation, because this will finally be very important for developing a causative therapy, because these entities behave completely different. And this is what, as Marlies said, led to this confusion, and also in the scientific community, whether tinnitus is now a more depressive state of the brain or whether it's a hyperexcitable state of the brain. And how the hyperacusis contributes to this conflict is what really is the most interesting idea of our current research.
Exactly. I must admit, sometimes that I'm writing papers and I have to discuss the mechanism of tinnitus and hyperacusis, when I get to some of the older studies and some of the newer studies as well, and then want to, and your own writings, and I want to interpret this in my discussions, it is quite a hard time. So I will be looking forward to listen to your presentations and learn from you in terms of how exactly these can be interpreted, and your research is quite translational. So you're over many years, in decades, you actually focused on this topic, learning from animal, now bringing the humans, and so it's a consistent process that it has to be to get to a place that we enhance our knowledge. Wibke, you also are involved in more animal side of the experiments?
Yeah, I'm more involved in the animal side of the experiments, exactly. And but as Lukas already also mentioned, for us is really the basic of the animal research is to do later translational studies also in patients. And we learned so much from these animal models the last years, and could really see that already on an animal model level, we have individual differences, and which of course is later also important to make human studies and to differentiate these subgroups, and that especially for this broad symptomatic diseases like hyperacusis or tinnitus is sometimes not so easy just to look, is there a hyperacusis or is there a tinnitus, but also relate these entities to other symptoms or other molecular markers. And there we learned a lot from animals, where we could differentiate subgroups and find different molecular markers and also different functional markers that now helps us a lot in translating these research to human beings.
And when you say molecular markers, are we talking about like things in a blood test, or?
For example, I mean, there's also, stress and tinnitus is not very new, but is still not really something that stress is very confused word that has a lot of interpretation. And I mean, we are now also starting to study how we can evaluate maybe from blood markers for stress levels, and have individual comparisons in animals that maybe will later also help to differentiate patients groups.
And then the functional markers is like MRI and
exactly.
The
Yeah,
electrophysiological tests and things
exactly, MRI, and also already from a normal hearing measurement, like an ABR measurement, you can learn a lot when you look a bit deeper than just to the threshold. You can interpret the different waves that reflect different parts of the auditory pathway. And this can be translated to humans. It's non-invasive, and there's a lot of things that can be analyzed in depth, much more than just are you have a proper hearing threshold or not.
Exactly, because this is actually one of the key priorities for tinnitus research, to find objective measures of tinnitus. And obviously the same problem is for hyperacusis, because we haven't got objective measure for hyperacusis.
Yeah.
And there the animal model gives a lot of possibility. We can do a lot of different measurements and try what measurements are effective to capture these differences in the animals that we can sort out already in the animal model, and then only use the ones that are effective later on in patient groups.
Fascinating. And there are several sessions in the conference, and one of the sessions is about psychological interventions for hyperacusis and misophonia, cognitive behavior therapy, dialectic behavior therapy, and also non-psychological interventions like auditory trainings and physiotherapy movements and occupational therapy interventions. And these are all at the infancy, if you like. They're not huge amount of clinical trials, but the people who be presenting there, they are in the planning stages for bigger interventional studies. And it would be great to have discussions and understand about your methods, and to see whether they can actually collaborate with you and bring some sort of an objective measures into the future clinical trials, which would be very valuable. Marlies, do you have anything to add?
Yes, this is the only thing. So in case that the time will allow, we may have also the chance to bridge a little bit, at least discuss with you and the others, because we also do EEG now. So this is something which is of course in the pipeline of being as a non-invasive but not so complicated measurements, possible to combine with cognitive training and to do this. And I look forward to discuss this with you and give you some insights, what we are planning and what we are doing, and perhaps even first results.
Fantastic. Thank you very much. Thank you for all of you to taking time and talking to me this morning. So hopefully see you on 1st of July in London.
Will be a pleasure.
Thank you.